So it's me again, Dr. Ranjit. So today what we are going to see today is about cell cycle and the pathway, the inhibitors involved, the regulators involved, and few MCQs high yield points regarding cell cycle. I'm sure all of you must have known about the normal cell cycle pathway, right? So I'm just going to start with the normal cell cycle pathway. You must have known about the phases of cell cycle, like we have a G0 phase, G1 phase, as in different phases of cell cycle. Generally, a cell cycle starts with a G1 phase. A cell cycle, the figure which you must have all known is a circle, and G0 phase is otherwise called as a resting phase of cell cycle. That's a resting phase where a non-dividing cell is going to be staying in that phase. So cell enters cell cycle either from G0 phase into G1 phase, or it can enter cell cycle immediately after mitosis without a rest. Can it enter into cell cycle immediately after mitosis? So from G1, right, it's G1 is also called as a pre-synthetic phase because this is before the DNA synthesis. It's pre-synthetic phase. After that it goes to S phase. S is your synthesis, where DNA synthesis or DNA duplication happens, and then we are going to have your G2 phase. G2 is pre-mitotic phase. Before mitosis we are going to have G2, and then we are going to have your M phase. M is your mitotic phase, where the cell is going to divide into two new cells. Then it can become two new cells going back to G0, or it can take a U-turn and go back to G1 again, right? That's how a normal cell cycle is going to happen. So I'm sure all of you must have known about this. So what you are going to see in this today's video is we are going to see about how a cell cycle is being regulated. The important questions in cell cycle come from cyclins, the order or the sequence of cyclins involved, and we have few pathways, we have few restriction points. For example, between G1 and S I have a checkpoint, between G2 and M I have a checkpoint. Who's going to control this checkpoint and how it's going to be controlled? That's what we are going to see today. Fine. So the first thing is going to be about cyclins. What propels a cell to go from G1 phase to S phase? What propels it to travel through the S phase, G2 phase as well as M phase, right? So we are going to deal about two different types of proteins here. The first proteins as group of proteins are your cyclins and CDK, which is going to be cyclin dependent kinases, and also we have your cell cycle inhibitors. Those are the cell cycle inhibitors are the one which are going to act on the restriction points and is going to stop the cell cycle. Fine. So let's see about cyclins first and cyclin dependent kinases, then to a cell cycle inhibitor. So as I said that G1 phase is where a cell is going to prepare for cell division. So it prepares for DNA synthesis. All the raw materials required for DNA synthesis is going to be taken care of during the G1 phase. The nucleotides, everything, the sugar molecules, everything required for a nucleotide is going to be taken care of by this stage. After this, it's going to go to S phase. S phase is where the DNA is going to divide. The DNA is going to duplicate, right? The DNA divides here, where we're going to have a DNA synthesis. We need a DNA synthesis here for the only reason that I'm going to divide into two new cells. I need two sets of DNA, right? So I'm going to have a DNA division here. Between this is our first checkpoint. In this checkpoint, there is a person called as retinoblastoma, RBG. Let's consider him as a watchman. So this watchman is not going to leave any cell from G1 to S. He's not a cell cycle inhibitor, he's a regulator, right? He's not going to send anyone from G1 to S. He's going to stop everyone. That's the role of every watchman, right? So now this watchman needs some ID card to go inside. Like if you go to a movie, you show the tickets, you are being sent inside. Same way here, I need an ID card. That ID card is your cyclins and cyclin dependent kinases. For my cell to travel through the G1 and S phase, I need two cyclins, cyclin D and cyclin E. We need two cyclins, and cyclin D has cyclin dependent kinase. It binds with the cyclin dependent kinase 4 and cyclin dependent kinase 6, and cyclin E binds with cyclin dependent kinase 2. I require these two. When these two are present, my cell says, okay, this is a kinase. Kinase, the role of any kinase is to phosphorylate a protein, right? That's what kinases do. So this is going to phosphorylate my retinoblastoma, and it's going to tell them that's a ticket for me. It's going to say, okay, I'm going to go to G1, from G1 to S phase, right? So if a question is asked, which of the cyclins is involved in G1 to S transition? So I have three of them. For an MCQ, you have to choose one, right? You're going to go with this combination. Cyclin dependent kinase 4 along with cyclin D is the most important cyclin required for a G1 to S transition. That's the first MCQ point here. Fine. And obviously the sequence can also be asked. The reason AIIMS exam, they have sequence pattern, so you have to know the sequence as well. Next, so I come to the S phase. With the help of the cyclins, we have reached the S phase. In S phase, my synthesis happens, my DNA divides. It's a semi-quantitative method of division. You will read them in your biochemistry. So everything in the DNA replication happens in the S phase. For my cell to go smoothly throughout the S phase also, I need cyclins, right? For my cell to go smoothly towards the S phase, it goes to G2. It goes G2 phase gradually. Because there is no transition here, from G2 phase, it's going to go to M phase. M is your mitotic phase. So, but here G2 to M, I have a checkpoint again. That's the second checkpoint, G2M cell cycle checkpoint. So, for a cell to smoothly travel through the S phase and G2 phase, I need cyclins as well here. So, here I need cyclin A with cyclin dependent kinase 2. That's required for me to smoothly go towards the S phase and G2 phase, and I have a transition here, I have a checkpoint here. This checkpoint, the G2M checkpoint, is being acted upon by two cyclins similar to my G1S checkpoint. Cyclin A with cyclin dependent kinase 2, cyclin dependent kinase 1, both of them help here, and cyclin B with cyclin dependent kinase 1, that's also going to help here. So here again, if a MCQ is asked, what do I choose? Because those are the points where we have confusion. We have confusion between two options, right? Here my answer is going to be cyclin dependent kinase 1 and cyclin B. That's more important for me for G2 to M transition. And for G1 to M transition, I need cyclin D with cyclin dependent kinase 4, right? So it has come to M phase. M phase, you must have learned from your 6th standard onwards, prophase, metaphase, telophase, anaphase. You remember that, right? That happens to mitosis and becomes 2 new cells. Okay, this becomes 2 new cells. This is how a normal cell cycle progresses. We have seen them vertically. We are not seeing them in the form of a circle, just we have seen them vertically. From G1 to M phase, we have seen them. Now, our question is, why do I need this guy? Why do I need the cell cycle restriction point? Why do I need a G1S gate? Why do I need a G2M gate? The reason is to prevent errors. When a cell is there, let's see our skin cell. You go out in the sun, you get exposed to a UV rays. UV rays is a mutation, is carcinogenic. UV rays can cause mutation. So this cell can already have a mutation. I should not let this mutation go to my daughter cell. That's where I have G1S. So G1S checkpoint is to look for pre-replicative errors. So the purpose of the checkpoint is what we are going to see here. I have a checkpoint. Why do I need the checkpoint? G1S checkpoint is to look for pre-replicative errors. So if there is any error, I'm going to stop the cell cycle saying, okay, you have a problem here. I have to repair the problem. Then only I am going to send it to S phase, because in S phase it's like a Xerox copy, makes 2 components. Correct or wrong, it will make it into 2 parts, right? So it's going to be pre-replicative errors. That's the first thing in G1S is going to check for. So here I need someone to stop that. If there is error, I need someone to stop that. That's where we are going to lead to cell cycle inhibitors. So what will happen to G2 to M? Here also I have, these are going to look for post-replicative errors. Okay, this is going to look for post-replicative errors. After DNA division, sometimes I can have a point mutation, one nucleotide A should combine to T. Sometimes it becomes wrong. That's an error. That can happen during DNA synthesis. That is going to be taken care of by G2M phase. So that's the reason we have checkpoints here. So if there is an error, I should stop the checkpoint. Who stops the checkpoints? Is our cell cycle inhibitors. These are going to stop the checkpoints, cell cycle inhibitors. We have a boss of all of them. We'll read them later on. So this cell cycle inhibitor, we are going to divide the cell cycle inhibitor for our ease of understanding into 2 groups. As usual, we have something called a specific, which is going to target few cyclins alone. Other one is going to be non-specific. Okay, specific and non-specific. It's a cell cycle inhibitor. In other words, they are cyclin dependent kinase inhibitors. Because cyclins regulate them, so I stop the cyclin, I will not be able to go forward. Right? Simple logic. So these are cyclin are cyclin dependent kinase inhibitors. So here, why I brought in this name is we are going to call them as CDKN family, cyclin dependent kinase inhibitor, right? CDKN. So the non-specific inhibitors are called as CDKN 1A family, 1B family, 1C, and so on. So specific cell cycle inhibitors is called as CDKN 2A, 2B, 2C, 2D, so on, right? So we have cell cycle inhibitors. They are cyclin dependent kinase inhibitors. That's it. So what are non-specific? Why do I call it non-specific? They can act on all cyclins. They can act on all cyclins or cyclin dependent kinases, A, E, or B, anything. There's no specific cyclin. All cyclins. Here they act specifically on cyclin D and your cyclin dependent kinase 4 or 6, right? It's going to act specifically on cyclin D. That's why it's called specific cell cycle inhibitors. Both of these are proteins. Everyone here in our body is made of proteins. So I'm going to give a term to them. I'm going to call them P and with a number. The number is nothing but a molecular weight. I'm sure you must have heard about the big boss P53. We're not going to discuss about him here. We'll discuss about him later, right? The big boss always need a special attention. So this non-specific cell cycle inhibitors, they're going to be P21, P27, P57. So P21 other name is CDKN1A, right? I'm just going to write it here so that you should not find it difficult when you have some big alphabet in the bracket. It's just an expansion, right? CDKN1A, 1B, and 1C, right? CDKN first group A, B, and C, and here we have P15, P16, P18, okay, and P19. So P is for the protein product and the numbers are the molecular weight, that's all. Here they are CDKN 2A, 2B, 2C, 2D, so on, right? That's it. So these are the group of inhibitors which is going to stop the cell cycle, which are going to act based on inputs which they get from different sources. So I have an error, my P53 detects it, says him stop the cell cycle. So these are the workers. They go there and stop the cell cycle and make sure my DNA repair mechanism is being recruited and they help in the repairing of the DNA. If possible, repair the cell. If not, kill the cell. My cell cycle division is perfect. It's perfectly regulated by different groups of proteins, cyclins as well as cyclin dependent kinases and your cell cycle inhibitors. So we'll stop the video with this. Okay, this is a short video just to give you a little bit insight about a cell cycle and where all questions can be asked. So let's go to few high yield points. Because most of the thing are MCQs. If we have the high yield point in the end, it's much more easier for us to revise as well. So the resting phase of cell cycle is G0. G0 is called the resting phase of cell cycle. Which phase is the point of entry into a cell cycle is G1. G0 to G1 is the point of entry of a cell into a cell cycle. Okay. We have two cell cycle restriction points, G1S and G2M. These are your cell cycle restriction points. Fine, they're just the cell cycle restriction points. We have read about them in detail just a few minutes back. So when I get a radiation, I have to kill a cell by radiation, right? Radiation therapy for a cancer cell. I can kill a cell only when it divides. So when I give a radiation, I have so many phases of cell cycle. Which phase a cell will be killed easily is your answer is your M phase. And a very important MCQ, M phase is the most radiosensitive phase. It's very sensitive to radiation. Because the reason is when a cell divides, all these components are going to be a bit fragile. A radiation comes and kills it very easily, right? Okay, and your G1S restriction point, you have your cyclin D with CDK4, and for G2M restriction point, you have your cyclin B with CDK1. Okay, and obviously you know the sequence of cyclins: cyclin D, cyclin E, cyclin A, cyclin B. This was asked in the previous AIIMS exam. The sequence of cyclins in the cell cycle, we have cyclin D, cyclin E, cyclin A, and then cyclin B. Okay, that's it regarding cell cycle. We have seen almost everything which is required for us for cell cycle. Let's meet you in one another video. Till then, bye bye and all the best from Dr. Anjit.